This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Europic! I am impressed. I thought you'd be silent. Good job. From Microbe TV, this is TWiV, This Week in Virology, episode 497, recorded on June 5th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today, we are recording at Europic 2018. This is a meeting going on in Egmond aan Zee in the Netherlands, and it's a meeting that happens every other year, and it's all about picornaviruses. And I have four guests from this meeting who we're going to chat with in the next hour. To my left, from Harvard Medical School, an old friend, Jim Hogle. Welcome.
Thanks, Vince. It's good to be here.
From Leiden University, Sasha Gorbalenya. Welcome to TWiV.
Yeah, it's great to be here.
From the University of Wisconsin-Madison, returning to TWiV for a second time, Anne Palmenberg. Welcome back.
Thank you very much.
And finally, from Utrecht University, Frank van Kuppeveld. Welcome and thank you.
Thank you.
I view this as publicizing and promoting your field, which is virology. That's why we've done this for 497 weeks. And today we have a little audience. So most of the people at this conference are not in the audience. They chose not to listen because they don't think it's important to communicate what they do. And I'm trying to change that slowly, week by week. Okay? But right now we have four people, and I want to find out first a little bit about your background. I'm really interested in why you became scientists. So let's start with Jim. Where are you from, and where did you go to college?
I'm originally from Minneapolis, Minnesota. I started at the University of Minnesota, and I went to graduate school at the University of Wisconsin.
And who was your mentor?
Matthias Sunderlingam, who is deceased in the tsunami a few years ago. I actually came to Wisconsin thinking I was going to do virology, and I originally actually talked a lot to Roland Rickert, and Roland wasn't available, so I was going to do a summer rotation with Sunderlingam, who's a crystallographer, and I just fell in love with crystallography and just never left. So.
And after that, you did a postdoc with
Steve Harrison at Harvard, and got involved in the early days when doing virus structures meant grams of viruses. And were plant viruses because they were much easier to grow and more stable.
And then you took your first job at Scripps, I remember.
Yep, and in La Jolla. And we started a new molecular biology department there. Richard Lerner put it together and put together a beginning crew of 2 crystallographers and a graphics expert, and then expanded and expanded and expanded. And the structure group just continued growing. And then about 9 years later, my wife had been in law school, and she was looking for jobs, and I had an offer from Harvard, and she had an offer from a couple law firms in the Boston area, and so we moved as a package. Went to Boston.
Yep. Yeah. Well, it's one of those cities where you can get 2 jobs.
Yes.
All right. But what really got you interested in science? Can you look back and say, I had a good teacher, or I read this book, or something like that? Yeah,
I kind of always interested in either being an inventor or a scientist. I think I had more aptitude for science and inventing, I guess. I had a couple of really good teachers that got me in. One was a teacher in high school, my biology teacher, that just was so excited about everything he was teaching that it was impossible not to get excited about it. And then I had a couple of really excellent professors at Wisconsin, especially my organic chemistry professor, and fortunate to have a biochemistry department at the time that I was in that really welcomed undergraduate students into the labs to do research. In fact, encouraged it. So the more I did research, the more I realized that's what I wanted to do for the rest of my life. And then I got enticed into doing administration instead. At the point, but I follow science vicariously.
That's the natural path.
Yeah.
You remember him at Wisconsin at all, Ann?
We didn't overlap.
Okay. Got it. Sasha, tell us where you grew up and went to college. Use the microphone, please.
Okay, so I was born in a city in Lysychansk, which was in Soviet Union and now in Ukraine. And I went to university in Siberia, in Novosibirsk, which is fresh new university, 10 years old, and was created to basically raise scientists. It is a very small university. And so I went from there, from Novosibirsk to Moscow, joined lab Vadim Hogle, and then I got in virology.
Mm-hmm. And did you do postdoc as well?
I don't remember.
Okay. Must have been memorable, huh?
Story was that I was in Vadim Hogle for training for 1 year or a couple of years, and so then Vadim accepted me as PhD student. Mm-hmm. But it was unusual arrangement because it was on the distant PhD student. I got my lab at age 25 in Siberia, and I flew to Vadim every 3 months for a couple of months working and went back. And so that was continued for PhD studies for 4 years.
So now you're at the University of Leiden. How did you get there? How long have you been there?
I was in Leiden University for 18 years.
Okay.
I joined in 2001. And my path was from Moscow, from Institute of Poliomyelitis, with Vadim Hogle lab. Mm-hmm. Through several sabbaticals, I joined Michael Rossmann lab for 1 year. I was sabbatical. Sabbatical was in Würzburg University with Stuart Siddell, and then I was visiting scientist in Frederick at National Cancer Institute campus for 4 years, and then I went from there to Leiden University.
No wonder you can't remember your postdoc.
Yeah.
If you look at his webpage, you'll see this movement all over, which is quite remarkable. Okay, and we've talked about your history a bit, but give us the summary. Where are you from originally?
So originally I'm from New York, and I did my undergraduate work in upstate New York at St. Lawrence University. And then I went out to Wisconsin for my graduate work, and I wanted to work with Sunderlingham and learn to be a crystallographer. And he told me that women couldn't do that because there was too much math involved. So I moved sideways and worked with Paul Caseberg on Q-beta polymerase and the biochemistry of that. That's how I got involved in viruses. And obviously we worked also with Roland Ruckert on picornaviruses and rhinoviruses and polio and so forth. And now that I'm back in that field, I now hire crystallographers as opposed to
women
as opposed to doing it myself. So fate takes very strange paths, but
Sure. So Anne was on TWiV. The name of her episode was "Wrong Form, Right Professor." And if you want to hear the story, listen to it. It's a really good story. Okay, Frank, where are you from?
I'm from a small village in the south of the Netherlands. You also asked what was the reason that you started to do science. I remember when I was in high school, I didn't like biology. In the end, I studied biology. But then we got the new insights about the chromosomes and the laws of Mendel, and that really attracted me. So I decided, okay, I want to do something with genetics. That's why I started studying biology. That was in Nijmegen, more in the east of the Netherlands. The first couple of years, I think they were terrible. There was a lot of botany and zoology and all this stuff, and we had to dissect birds and all of the stuff, which is not my case. But I hang on, and then only after the third year, when the molecular topics came in, then I could really expand myself and flourish. And that was it. Then I graduated I did a major internship in virology, and that really stuck me. So I did a PhD in virology. I was very much interested in membrane transport. So I did one postdoc outside the field of virology, working on regulated secretion and how that regulates secretion. And then I went back into the field of virology and never left it. And I stayed for a very long time in Nijmegen. And only 6 years ago, 2012, I left Nijmegen to take up the chair of the University of Utrecht, the virology department. Mm-hmm. And there I'm now in a very big department where we not only work on picornaviruses, but mainly corona and flu. We didn't work before in Utrecht on picornaviruses. I took my whole group with me, and so...
Got it.
That's how it is.
All right. So, Anne, I forgot to ask you, what got you interested in science?
I got interested in science in high school biology.
Okay.
I distinctly remember the lecture of DNA to RNA to protein, and that's when it occurred to me that there was a genetic code that explained all of life. That all of biology basically was encoded in the genetics of nucleic acids. And so fundamentally the study of nucleic acids and how information is encoded in that, I got hooked in high school.
Got it. Sasha, do you remember what got you excited about science?
Oh, yes. Also the same. Basically, it is teachers at the high school.
Okay. Yeah, me too. I had a great high school biology teacher. Now, this meeting, Europic, Frank, do you have some idea of the history of it? Why was it started?
I think it was started, and I heard it this morning. I think Eckhart told me it's somewhere in the late 70s. The initial meeting, I think only Europeans were allowed, therefore also the name Europic. They didn't want these arrogant, big-mouth Americans on board. That's how it all started. And started off, I think I once heard the first meeting was with something like 20 or 30 people. Mm-hmm. And I even, Dave even once told me, Dave Rowlands, that I think you went to the first or the second meeting. Yes. And you were in a room together with someone else, and you even had to share the bed because it was a very low-key meeting. And that then, and that over time it expanded, and my first Europic was in '94 in Helsinki.
So you remember your first one?
Oh, I remember it very well.
Do you remember what you presented?
Excuse me?
Do you remember what you presented at that first Europic?
I don't know. No, I don't remember.
No one can remember. Okay. I can you remember? When was your first Europic, Jim?
1985.
Ah, yes, I remember that one. That was at SAAC, right?
That was at SAAC.
What did you present?
At the poliovirus structure for the first time.
Yeah,
it was actually the first time I ever presented it outside of Scripps.
Right. And, you worked up to that. You made sure it was done by then, right? I remember.
There were other reasons for making sure it was done. It was we were in the midst of pushing the publications out while I was there, but it was also a very exciting time because of the nature of the fitting the job into small computers. I'd not been sleeping very much, and so it didn't bother me not to sleep very much at that meeting. I can't do that anymore.
I remember SAAC very well. Michael Rossman had come with the rhino structure, and he had a little wire frame protomer, BP1, 2, 3, and 4, which he carried around all the time at the meeting. And you could ask him, where is this amino acid? And he would point it out to you. It was very exciting. Sasha, when was your first Europic?
My first was in Gmunden in Austria in 1996, but I don't remember what I presented.
Okay. So, Anne, you remember your first Europic?
I think the one before SAAC was in Rome.
Urbino.
And I remember talking about the 3C protease that we had, Mark Palanch, who's out here somewhere. Mark was a grad student with Roland, and I was a postdoc, and we had discovered, by my mistake in a lab, what the 3C protease was and where it mapped, and that it was responsible for doing all the polyprotein cleavages. Mm-hmm. And I think that was presented at that meeting.
So my first one was Urbino, 1983. I had just gone to Colombia, and I got a call from Raul Perez Berkhoff.
Oh.
A few of you remember him, but not the young ones, not at all. And he called me in New York. There's a day when you used to make phone calls. And he said, you have to come to this meeting. I said, what is it? He said, well, it's the European picornavirus meeting. I said, okay, but I don't have anything to present. He said, that's all right, come anyway. But we had just made an infectious clone of polio, but I didn't think that was enough to present at this meeting. But I do remember at the meeting, Roland and Eckhart presented the 434 nomenclature for the picorna genome. That was the first time we'd heard of it. But really small rooms, with 20 people in them, nothing like this with thousands of them right now. All right, so one of the topics of this meeting has been the eradication of poliovirus. Not poliomyelitis, poliovirus. So I want to ask you all a couple of quick questions. Yes or no? Jim, will we eradicate poliovirus? Yes or no?
Yes.
Sasha, yes or no?
No.
Anne, yes or no?
Yes, burn it.
Frank?
No.
No.
Can I qualify my answer?
Good.
I think we will succeed in eliminating the current viruses at some point. It may not be nearly as soon as other people think it's going to be, but I don't think it's going to go away. I think that there will always be something that causes poliomyelitis.
Okay. Now another question. Let's assume we do eradicate poliovirus. Frank, should we stop vaccination at some point?
No.
Anne?
No.
Sasha?
No.
No.
Wow. Pretty good. Pretty interesting. Nobody cares what I think, so that's fine.
We know what you think.
So
Yeah, just so you know what I think. It's very interesting because the session kind of presaged the first and second answers. So no stopping of vaccination is very interesting. So Mark Planche, listen to that. All right, let's talk a little bit about science. Sasha, I was told that you recently retired. Is that true?
yes.
Now, what does that mean here in the Netherlands? Do you still go to work and do the normal things? They call you retired? Or?
In this country, it is very important balance of life and family life. So you're not expected to go to the office.
You can't go to the office at all.
You don't have office.
Okay, and so what do? You come to meetings.
I'm still part-time employed.
Part-time.
This year, right. So I still have office, but probably the last month.
And then you won't have an office after that.
Yes.
But you can work. So most of your career you have done computational biology. Would that be fair to say?
Yes.
That's a term that Eugene Koonin said to use, because I used to use bioinformatics. He said, no. Computational biology. So you can do that at home, right?
Yes.
Okay. So I think in your early days, you did a lot of work with Eugene Koonin, right?
Yes.
And you published a lot of papers on motifs. These are the early days, right? Early 80s. Proteases, polymerases, helicases. And I think many of us probably remember those early papers that inspired biological experiments. Where did that come from? Where did your interest in looking at proteins in rudimentary computers, where did that come from?
So this was story when I did my PhD.
Yeah.
It was, I wrote my PhD thesis in Siberia. And for my kind of review in PhD thesis, I just wanted to understand my data better experimentally. And so at that time, Eckhart Wimmer published full genome sequence of poliovirus, and I was working on cardiovirus. And like N in parallel, I mapped protease on the genome, but I did not understand what type of enzymes is, class enzyme. I knew catalytic residue, but I did not know sequence. So I decided to look in the sequence Eckhart released and to map enzyme. Mm-hmm. So but there was no bioinformatics. But basically asking questions there. So we started bioinformatics, and so we were able to map. And so actually we compared your sequence with Eckhart sequence. We found that Eckhart sequence was wrong in certain places. Sorry, Eckhart, but it was important, and exactly in the place where it was very important. So your sequence and Eckhart sequence very help us to answer question about catalysis, about motifs, and that it was started in Siberia. So then I went in Moscow, and we joined. Eugene joined the group, and so we started.
And do you remember some of his early work? Did it influence you in some way?
Oh, we used to compete. I can remember when we were working on the sequence of EMCV, which was new at that time. And I got a phone call in my office, and it was Vadim Hogle on the phone from Moscow. And this is before you could have phone calls to Moscow. And he says, I need your sequence. Okay. Okay. And he was sequencing proteins, and we were sequencing RNA. So I read to him over the telephone to Moscow the sequence of the EMCV polyprotein. Because it would have taken months to send mail back and forth. And I repeated it twice, methionine, alanine, threonine, and the entire genome, right? And 2 days later, the CIA were in my office. What is this code you are doing with Moscow, So Vadim and I, because the Russian groups were working on cardios, as were we, and that was the main way to exchange information. We competed with each other on proteases and on sequences and leader proteins, and it became we're best buddies. We're best buddies. It became much easier to collaborate than to compete.
Right. So now
you didn't tell the answer that you gave to the CIA when they asked you about the code.
Well, this is this is sanitized. Let's not go there. But when I go through passport control, okay, there's still people who kind of go, oh, really?
If you did that today with certain sequences, you would get flagged also.
Yeah. Well, it was. I mean, obviously, all the lines were bugged, and you could hear people clicking in as you're reading single letter amino acid code over the telephone.
By the way, I just remembered that Ann and I have the same license plate. She lives in Wisconsin, and I live in New Jersey, but we have the same license plate. What do you think it is? Viruses. And someday we're going to drive to an intermediate place and take our pictures together, right? One day we're going to do that. Sasha, let's get back to you. So how would you say over the years, from those beginnings with motifs, how has your work evolved?
So, as I said, I started as experimental virologist. And so basically, bioinformatics, its motifs, it came as a result that it could be insightful. And so because of my origin as experimental biologist, so I was concerned about this link between experimental research and theoretical research. What you found when you're doing bioinformatics, it is a really big insight, but it is on the border that it looks like unbelievable. So it's very difficult to convince people that it's something that it's true. So my basically concern and my drive was subsequently was to collaborate with people and to help people formulate questions and also analyze questions. So this is why I stayed in experimental labs and departments. So to be on the connection between the bioinformatics and experimental research.
And that's why eventually you moved into other viruses like coronaviruses as well, right?
Yes. So in the earliest in 80s, it's probably the best time in virology in terms that it was genomics started. And so all virus families, first genomes released. So you could look at viruses and you could tell what viruses is about. And in many cases, like in coronaviruses, molecular biology was in nascent state, so it was nothing. And so we were able, first time, to basically to say coronavirologists what is you could expect. And so it was determined for them for 10, 15 years what to prove. So that was...
And this, you can continue to do this from your home, right?
Not nowadays. It's science, of course, moved. Of course, it is different scale now.
But if someone called you and said, "Can you look at this sequence and tell me what's in it?" you could do that.
Yeah, certain sequences and certain questions.
Okay. Okay. The other thing you've been interested in is viral taxonomy, right?
Yes.
Why is it important to have a viral taxonomy?
Okay, so why taxonomy generally important? So you would like to have certain order in the data. So in biological world, outside virology, taxonomy emerged before evolutionary biology. So in here, it is reverse. So taxonomy it is very young discipline in virology. Like virology is very young. So you would like to make order. So, and best order would be evolutionary order. So then if you have certain type of order, you understand what you deal with. And maybe we as a biologist will start studying not simply just individuums, but species, genera, and families. We don't do this. We don't have, for instance, studies on species level. Virology is not recognized.
Okay, so if you were not retired, what would you do?
I'm still not retired completely. So, and I don't know what would happen. So first I would like to sleep for for months, probably for 2 months, maybe 3 months. And so then I would have clear mind and I will tell you what I'm going to do.
Okay. I want to ask you a very specific question. What is the upper limit to RNA virus genome size?
Okay. Okay. So we know probably a chemical limit of RNA size. It is pre-mRNA. So this would be kind of chemical limit. It would be probably up to 200,000 nucleotides.
Okay.
So, but we don't, of course, know what would possible to replicate, and so in viruses. And so this is what we expect, at least, and hope when continuing characterization of viruses or diversity of viruses, we could learn this limit. We don't have idea, we don't have theory which would explain upper limit of RNA life.
What's the biggest virus genome we know of right now?
So right now in the bioRxiv released a study which I'm kind of co-author, it's 41 kilobases RNA genome.
Okay.
So this is current limit. So, and that next virus, smaller than this virus, by poliovirus genome size.
Okay.
Yeah. So, and probably it is not the limit.
Yeah, we don't have everything cataloged that's out there for sure.
Right. Yeah. But what is important, if you kind of could spend 1 minute on this. So, all our analysis before, sampling showed that we already reached limit.
Right.
Because change of genome size along with our characterization of virome did not change much. So it looks like limit. So now suddenly we see increase in genome size in this virus particularly. And so there are some kind of some changes in organization of genome which could explain partially why it happens. But we don't have mechanistical understanding constraint on genome size. And so because of this, we have poor theory what to expect.
Okay. I think we're going to still see you on papers for the next 20 years, so whether you're retired or not. And one of the things you're going to talk about at this meeting is women in science. Tell us what you're going to say.
I'm going to say what Frank told me to say. I came here, I was going to talk about structure. Yeah, well, I'm senior enough now in my career that I can do political things as well as scientific ones. And a couple of years ago, as part of ASV and then getting involved with some of the other virologists at Wisconsin, we started putting together lists of how women are used, shall we say, in scientific conferences. And it was actually quite a simple thing to go through program books and see the frequency with which females are invited or not invited to major scientific conferences. And we wrote a paper, Rob Kaleda and I wrote a paper on that, and the findings were rather, they were shocking to us and they were shocking to the people who read them. Basically, what criteria are used to select symposia speakers at major virology conferences? And there's a certain bias involved, and the bias has to deal with picking, people who have been there before as opposed to youngsters, or just the gender of the organizers themselves, who tend to pick friends as opposed to the best science. And one wants to put the best science in front of an audience, but in fact that's done by going, oh yeah, but my buddy, he's got a really cool new paper, all right? And when you do that, you don't seem to see female faces on the podium. So by writing that paper and looking at the parameters that went into the selection of males and females at major virology conferences, it's opened a lot of eyes. It certainly did at ASV, and it has at the Plus Strand. And I gave Frank here a copy of that paper before we published it, and he saw the light, okay, and became a convert, and it's made a difference at Europic, and I think it'll make a difference at the next Plus Strand as well. So I can come to any of these meetings and I can talk about science. I always have wonderful stories to tell. I love telling them. But he sort of convinced me that it would be very useful to stand in front of an audience and share what we learned by doing that paper. And my experiences as a woman coming up through science, I'm not a crystallographer, and in retrospect, that might be a good thing. I think Sunderlingham did me a favor in the long run. But career choices for women and minorities have been limited by the opportunities that are presented to them, and I'm aware of that. And I can stand up and, if nothing else, perhaps open eyes, and that may change the way the future generations have different opportunities. And if that's the case, I have no problem standing in front of an audience and telling them what we learned. So that's what we're going to talk about tomorrow.
Do you think such studies will have an impact?
Already have.
How so?
When I was president of ASV, I did the first. I went through. The job of the president of ASV is to put together a plenary slate. Okay, and I went to the ASV and I said, "Who were the previous speakers, so I know not to pick them again?" And they said, "We have no idea. Why wouldn't you pick them again?" I was like, excuse me? So I pulled out the program books and just made a list. It took a day and a half, maybe at most. And I was astonished to find that in the previous history of ASV, for the first 30 years, 85% of the speakers were male. Okay. And the demographics were 45% women at the meetings, and yet 85% of the speakers were male. So I changed that in my year, and I went to the council and I said, and I did that because here's the list, here's your history. Is this the history you are proud of? And they didn't answer that question, but the spreadsheets became used by the next series of presidents. You in particular were a couple years after that, so that they simply could look at these spreadsheets and go, oh wow, I hadn't realized this person has already spoken 6 times. Maybe we can find a fresh voice. So it just changed the perspective of who they were looking for and trying to deliberately put more balance into programs. So it shifted ASV within 10 years from 85% male to about 60% now. In some years, it's even better. So it made an immediate impact. 2 years ago, we did the same thing for the herpes people, who were 95% male, okay? And now they are 85%.
Well, they're a big virus that's slow for them to turn.
They're also herpes. I mean, people who go into that field, you have to question what they do anyway. But So it has made an impact, and I have people come up to me at meetings, especially young women, who go, "Thank you for writing that paper." Okay.
So, will there become a time when you don't have to keep reminding people, when it just happens organically, 50-50?
Never. Never. But what it does is provide another level of transparency. Just as in your science. I mean, you go to a meeting because you stand up and put your science and people talk about it and do things. You learn from experience, and you learn from what other people are doing. And if someone stands up and says, if you're going to organize a meeting, you need to be aware of these parameters. Mm-hmm. Then when you organize a meeting, you become aware of those parameters. So it's a learning process. It's a continually learning process. Transparency is the key.
And everyone has to keep bringing it up. Let's keep it balanced in all aspects. And hopefully it will move to the larger scientific platform as well. Other meetings, big national, international meetings, publishing, all of that.
I actually heard a rumor that the HIV people are going to do this now. I'll believe that when I see it. Okay. But
I'm waiting for Congress to be 50-50. We can do it. more women running this fall than in the U.S. than
Why
any other time.
50-50 and not 60-40?
Okay. That's okay. We could do that. We
We'll talk about that tomorrow.
We could do.
We'll talk tomorrow about the difference between equality and equity.
Yeah, whatever you want. Actually, it should be whatever it ends up at. Unfortunately, previously it ended up at a bad place because people weren't talking about it. There was no transparency. So you can't talk about science tomorrow? Frank's not going to let you? Is that right?
That's correct.
So tell us right now what's hot and exciting in your lab.
We've got the CDHR3 at 5 angstroms.
And I'm sure all our listeners will get that.
We're doing the receptor of rhinovirus C. You heard some of it this morning from Kelly. That work is the tip of an iceberg of more than 4 years of biochemistry in our lab to get to the point where we can do the soluble receptor bound to rhinovirus C. And that's a huge breakthrough in asthma research because of what rhinovirus C does. And as we are doing this, we're designing the drugs that will inhibit this and help prevent asthma.
Well, there's a field that's changed substantially, the structure field, right? I mean, we hear huge 1.8 angstrom resolution from cryo-EM now, when that was only possible with X-ray crystallography. It's amazing.
Right. So I have figured out instead of doing the structures myself, I get someone else to do them, and they give me back the PDB files to do my biology.
Right.
So I'm a closet structural biologist.
Well, we could talk more, but let's get to some others here. Let's get to Jim, and if we have some time, we only have an hour here. Now, Jim, I remember I was a postdoc, and you called me one day, and you said, I'm working on the polio structure. This was probably 1980, 1981.
Yep.
And I need the sequence, and I had to give it to you on a computer tape. Remember, a big reel? People aren't laughing because they don't know what computer tape is. It was this huge reel, which I had to go to the computer center, and you said, we'll probably get it in about 10 years.
That was the schedule.
So why was it so accelerated? So you reported it in SAACS.
Things worked out. That plus the fact, it's amazing how fast you can run when you're on fire. And it was really clear that Michael Rossman was working on rhinovirus, and this is a very comfortable relationship in a way for a while because we thought they were going to be very distantly related viruses. And then when the Rhino 14 sequence came out, and we did a little Sasha gazing, we realized that we were talking about very close cousins. and it was just, the point was not to be way, way, way behind because part of it's because I was a very competitive guy at the time. The other one is Scripps had made a huge investment in us at the time, and my work was in the structure group at the time was really the only thing that was going well. And so the idea of getting something out there was very important.
So after you had that initial structure, that really drove your lab for the next 20 years, right? My initial interest in the field was I was just crazy about things that self-assembled and how that worked. And so,
after the virus structure, we do another serotype, and then that gets to be things that you could begin to predict, with at least some confidence what the next serotype is going to look like. And then we tried to do work on lancing for a while because we were interested also in the what changed the tropism of a poliovirus so it was able to infect mice. But it was just this really fundamental interest in assembly. So we worked in assembly for a while, and then got distracted and started doing entry. And that was about the time, a few years before I left Scripps. The last few years I was at Scripps. But it has really been what's pushed my work since going to Harvard, and that was 27 years ago. So,
so you continue to work on entry, right?
Yeah, and it was one of these things where I burned a couple of postdocs trying to make crystals of the known soluble forms of the entry intermediates, and succeeded with both, but the A particle crystals were not usable. the heat virus crystals were beautiful and diffracted to 9 angstroms and were perfectly twinned, which means you can't solve them unless you know the structure. And so I was kind of dragged kicking and screaming into electron microscopy. And for a while we were collaborating with Alistair Stevens' lab. And then it got to the point with some of our samples, we needed to be hands-on, direct from the bench to the microscope, just to be able to figure out how to make the samples that we wanted to work on.
And that was a collaboration we had with polio and polio receptor.
Yep, that was part of that.
Which is on my cell phone now. The cover of my phone is the polio receptor structure.
Yep.
Do you still have unanswered questions with polio assembly and entry that you think you might not be able to answer before you have to stop?
I actually, one of the real fundamental things that we're interested in right now is what the machinery, our model, I think is gaining some acceptance, is that at least with most of the enteroviruses, the virus makes a pore through an endosomal membrane that the RNA is translocated across. And we have some experimental models that suggest that this works, at least in experimental models in vitro. I want to know what that machinery looks like, and this is pushing some things that, Susan's doing, Hafenstein's doing some very similar things. It's pushing you into getting away from the comfort zone of having to make use of this symmetry, which is a godsend for doing either crystallography or electron microscopy, and trying to have to do asymmetric structures, which means you have to have a lot more data in order to come up with a reliable structure. But we're working on several structures that are trying to fill us in on what the translocation machinery looks like. And then, of course, then the questions come up is, yes, but how does it really get set up, and how does it really work? But I came from a background. I think the person that said it directly, that really hit me was Don Wiley, who said, I don't really understand anything unless I can see it. And so I was late into sequence gazing because I'd like to see in three dimensions. But man, there's nothing like taking the structure of, a reasonably high resolution structure of the virus and then going back to the sequence and being able to just build the sequence into real electron density and take a one-dimensional sequence and put it into 3 dimensions and begin to try to make sense out of what everybody else in the field was saying based on sequence information and genetics. Yeah.
So your main focus is still poliovirus, correct?
We've done some. I have a part of the lab that does work on herpes proteins. We've done the processivity factors. We've done the nuclear egress complex. That's a small part of the group. The main part of the group's always been working on polio. We digressed for a bit. We did Tyler's. We did some cardioviruses. And at about the time people start threatening whether we were going to be able to continue to work on polio, we jumped into Echovirus 1, only to discover that it was very frustrating because what you're going to either go back and say this looks exactly like polio, in which case that's boring, or you're going to go back and say this is completely different, in which case somebody may not believe you. But the idea of what's kept me in polio is just, I think, the wealth of the material that was available when I first started, and just the activities of the people in the field that just, we know a lot about this virus that we might have in working in a less understood system, have to just recapitulate all the experimental work, and getting to the point where there aren't a lot of people that want to do all the experimental work with polio anymore. Or
can't eventually,
yes.
Right? What is your favorite paper that you've published?
That's the 1985 science paper. It was just an experience of getting into that work. Was literally, I was working with a senior postdoc in my lab. Every day we were trying to fix the rate limiting step in doing the computations, and we would kick around some ideas and go back. I'd get in at work early and I'd be working, collecting data, and then he'd come in and we'd sit down and we'd talk. And he said, well, I sketched out this idea and this idea. They're not going to work. I did this one. I think it's going to work. I'll have the program built maybe tomorrow. And, that was every day for like 6 months. Then you put the paper together, and it turns out that the structure immediately had nice stories about antigenic sites and some hints about assembly in that. And it's just very exciting.
Sasha, do you have a favorite paper?
Yeah, I just was also thinking about your question. And, it's like about your children. So you ask about who you love most. I mean, you cannot answer this question. So, and also that some children kind of succeed. And so they kind of, and some not, but that quality is there. And so, of course, there are different stories, like Jim said. So with every child, you have a story. So, yeah, I'm kind of fortunate. I have family.
Okay. you love all of your papers.
Yeah, I love. Yeah.
I know it's a hard question, and sometimes in the audience or people from your lab, you don't want to insult them either, right? So, but nevertheless, most people find an answer. Do you have a favorite paper, Anne?
I don't have children, but I have papers. I think one of my favorite ones was in 2009 when we sequenced all of the rhinoviruses, mostly because that indulged my love of sequences and bioinformatics. And that was a lot of fun to put together, and it got a big publicity splash. That had 90 new genome sequences in it, and we were very, very proud of that. And 2 years later, we put out 240 more sequences, and they made genome announcements, and that was it.
Yeah.
So it sort of keeps you humble.
You can't publish sequences anymore.
That's right.
We in Eckhart published a polio sequence years ago. It's a Nature paper, PNAS paper. Forget it. It took us years to do that. Now you mail it away and it's done. I have to tell you that Anne made me a poliovirus with the genome inside beaded. Remember?
Sure.
7,442 bases. Each of the 4 bases is a different color. And she threaded it. You said while you watched the World Cup, you told me.
Football games.
Football game.
Halftime at the football game.
And so it's stuffed inside a capsule. You can pull it out, and it's roughly to scale. And it's a wonderful teaching tool. But thank you for making that. I love it. It sits on my desk.
I can probably recite most of those nucleotides.
I'm sure you. Russian? Yeah. All right, Frank.
I think my favorite paper was the paper that I published when I was a PhD student. I was working on the TWiV protein, and we showed that was some kind of a viroporin, and that it targeted calcium and Golgi membranes and the filling state of the ER and the Golgi, the calcium filling state. Meaning we used all kind of fancy probes, equorins that we targeted to those compartments, and we could really show how infection and the TWiV protein affected the filling state. And I also remember that paper was rejected by EMBO Journal. So, and I was very mad about it. So I took the phone and I called the editor-in-chief, Ian Mackay, and I explained them why they made a mistake. And then at the end of the phone call, he said, send it again. And it got published.
Well, I think that's a good recommendation because we don't use phones anymore.
Yeah.
But maybe you should still call
I
editors.
also had a point. So
yeah, I think
that's not the way you felt when you were editor. I mean, when Vince was editor of JViral, and I wanted to talk to him, he was basically pre-screened all the calls because he was having all these irate phone calls from people that weren't happy with their reviews. And I had to assure him I had something interesting to talk about. Sometimes I was complaining about the reviews instead. I fooled him, but
I don't remember that at all. But those of you who are younger and want to publish papers in, quote, high-profile journals, I would say, call the editor and complain, because that really works. I don't do it, but a lot of people do, and if you don't make noise, no one's going to hear you, right?
I never did it again.
Although
it was successful, I never did it again.
Now, you started working on picornaviruses, but then you also included coronaviruses, nidovirales, right? Why was that? What got you interested in that?
Well, that was mainly because that was because of my move to Utrecht, and we are working in the picorna field, that is on replication and replication organelles, and also how picornaviruses interfere with stress and with interferon signaling. Those kind of things. And from that perspective, also, we found some very interesting coronavirus proteins that also could have an effect on formation of stress granules which have some effect on translation initiation. So it came from the link of innate immunity and the connection with the coronavirus group in the Utrecht.
Got it. In fact, I saw you and Sasha at a nidovirus meeting last summer. So here you are, you dip into both pots, picornas and nidovirales. You went as well, Jan, yeah. But you don't do any corona research, right?
I was the token female.
Actually, it is also very interesting because this connection between picornavirus and nidoviruses was started through bioinformatics. So when first genome of coronavirus released and we analyzed them, we found that coronaviruses use 3C proteases, and so processing is like in picornaviruses. So we transferred knowledge from picornaviruses to nidoviruses, and then it's a mutual benefit. So it was also...
Got it. Makes sense, yeah. Now, Frank, what's exciting in your lab right now?
What's exciting? I think we have some very nice collaborations so that we can also use a lot of advanced microscopy. I mean, you talked about the advances in structural biology and cryo-EM, and I think also the use of CLEM, correlative light electron microscopy. Tomorrow the audience will hear a talk about serial block phase scanning electron microscopy, so you can now go through an entire cell and assemble all the pictures that you get. So to get a real comprehensive picture of what's going on in an infected cell. We are setting up also some microscopy, and it seems very promising to visualize translation. So we can visualize translation of the incoming RNA, and hopefully soon we will also be able to visualize the RNA itself. So I think also the advancements in microscopy will have a big impact in all the research that we are doing.
So how has this meeting changed from the first time you went to today?
For me, it didn't change at all.
Exactly the same.
It's the same. Yes, yeah. For me, it's the same. Yeah.
What about you, Anne?
It's still poliocentric.
So it hasn't changed. You see no changes.
It's broader. There's much broader base,
Really?
but there were whole meetings where there would be maybe 2 talks that were not polio.
Sasha?
Yeah, I agree. I mean, there is change, of course, in topics, and also you could see friends like you graying. Yeah, so
Graying. Yeah.
Yeah, so there are changes. Yeah, there are changes, but there are also some things that you love to come.
Jim?
Well, I think the sense of community is one of the nicest things about these meetings. I do dabble in the herpes field, and it's an entire different story going to a herpes virus meeting and watching people clobber each other all the time about minor points, and especially when it's the senior people clobbering the young people out talking. And I just that's not a very. This is a much more comfortable field. People actually get a chance to like each other most of the time, which makes it nice. But one of the changes that I have seen is that. There was a time when I was on most of the programs, and it gets to the point where the first couple you really need the exposure, and then after that it's just nicer to listen. And I think we're seeing many more young people talking at the Europic meetings than it used to be. It used to be fairly senior people that were giving the talks, and now it's more and more students and postdocs that are getting a chance to say something, which I think is a really positive change.
I have a good friend who's a pox virologist, and every year a bunch of them go golfing together. It's called the Poxvirus Invitational Tournament. And they have said to me, the coronavirologists could never golf. They don't like each other.
Well, I can't golf. That has nothing to do with liking.
I just want you to know how some other virus feels about us. So you might be right, but and I think you are right. There's a wonderful sense at these meetings, but maybe the outside world doesn't know it. I have one more question for all of you. Let's start with Frank. If you had not been a scientist, what would you have become?
A football reporter.
A football reporter?
Yeah. Or a journalist. Journalist, maybe.
Yeah. Would you be a science journalist, you think? Or straight news or sports?
Well, maybe sports journalist.
Do you like sports?
Yeah, I like very much. Okay.
And what about you? What would you have been if you hadn't been a scientist?
I'd probably be married and have 4 kids and drive them to soccer practice.
Well, that's a job, right? It's a career. Sasha.
I probably could not change. Yeah.
You would always want to be a scientist.
Yeah, in one way or another. So maybe a different field, maybe, yeah. So.
Okay. And Jim, what would you?
I can't really imagine doing anything else. I just, especially in the early days when it was a small group and I was in the lab, and people get scared when I threaten to go back in the lab now. But when I was just really doing research, and that was everything I was doing, and I kept coming in every day and saying, what's wrong here? People are paying me to do my hobby, which, usually being paid to do your hobby ruins it, but not doing science. It was just, I enjoyed it every day. Administration, less so, but...
Well, when we do TWiVs on the road like this, I often ask my guests that question. And so if you want to hear some other answers, you should listen to some of the old ones. There are some very interesting answers, some interesting alternative careers that people have. But if you ask me, I would say I always wanted to be a scientist. And I think that is the requirement, in a way, to be one. David Baltimore once said to me, if you can think of doing something else in your career besides science, do it. There you go. Well, that is, in a nutshell, the first TWiV from Europic. Maybe it won't be the last. By the way, I started TWiV 10 years ago, and that was the last time I came to a Europic, which was in Sitges, right? And at the time, I was thinking about the podcast already. And in the intervening 10 years, I did it. Most of the time we talk about papers. I have a cadre of hosts who get on Skype with me, and we talk about papers. And the podcasts have expanded to other fields as well: microbiology, parasitism, evolution, immunology, and each has a separate set of hosts. And the goal is simple: to tell the public what we are doing, because they don't know, and we are the ones to tell them, because we have the passion and the understanding that no one else does. So you can check out some of our other work at microbe.tv. If you have a phone or a tablet, you have podcast players that you can use to play podcasts. You could just subscribe. It's free. We do all this for free. You can subscribe to TWiV or TWIM or TWIP or TWiVo or Immune. Those are our main podcasts. And on every podcast, we get questions and comments, which is really a lot of fun from listeners. 70% of our listeners are not scientists, and I think that's pretty good. And for this podcast alone, about 20,000 people are listening. And my goal in the rest of my life is to make it 10 times more, because I think it's really important to do that. And I need your help too, so when I ask you to do one, don't say no. Anyway, you can send your questions and comments to TWiV at microbe.tv, and if you like what we do, consider supporting us financially. We do this for free, but we do have expenses, and when we travel, it costs money. You can go to microbe.tv/contribute and find out ways. For example, you could subscribe for $1 a month. Today's guest, I want to thank, from Harvard Medical School, Jim Hogle. Thank you very much for joining us.
Thanks for inviting me, Vince.
My pleasure.
Not that you gave me any choice. So. Vincent, but he called. He wrote me one day, and I hadn't answered yet. He wrote me back 24 hours later. It's like, well,
yeah, you have to answer, because it's important to do it. We only touch the surface here. But if you go, for example, to Anne's TWiV, we get deep into the science. I'm not afraid of doing long form. We usually do 1.5 to 2 hours per episode. And can you imagine that 20,000 people are listening to that? It just goes to show that 20-second journalism isn't the way to go. From the University of Leiden, Sasha Gorbalenya, thank you.
Yeah, thank you for the privilege.
My pleasure. From the University of Wisconsin-Madison, Anne Palmenberg.
Thank you very much, Vinny.
I think it won't be your last. And from Utrecht University, Frank van Kuppeveld, thank you.
Thank you, and thank you for coming to the Europic.
Well, thank you for inviting me. This was your idea last summer at the Nido virus meeting. You said we should do this, and I appreciate it. I appreciate your invitation, all the organizers inviting us to do this, and I appreciate the audience to come here. I'm Vincent Racaniello. You can find me at virology.ws. We thank ASM for their support, and Ronald Jenkins for the music you hear on TWiV. He's at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.